Hello, and welcome to my protocol video on how to do a yeast DNA extraction with no phenol, no chloroform, and for a fraction of the cost of a kit. My name is Julian Lieber. We're going to start here with a culture in YPD medium. Vortex the culture to resuspend cells. The yeast I'm using is called Basidium pullulans, but this protocol should work for most yeasts having cell walls that are digestible with the enzyme Lyticase. This protocol is adapted from the Promega Wizard genomic DNA kit. Aliquot 1.5 milliliters of suspension into a 1.5 milliliter tube. This will be for extraction. I also prefer to make a glycerol stock of the same culture to ensure that any DNA sequences detected in the extract are the same as in the archival sample. To make the glycerol stock, add equal parts of sterile 50% glycerol and cell suspension, then mix and freeze for long term at -70 to -80 Celsius. Centrifuge at 16,000 RCF for 1 minute to pellet the cells. All subsequent centrifugation will be at the same acceleration. Pour off the supernatant. In order to degrade the cell walls of our yeast, we're going to add Lyticase. I had previously made a stock solution of 672 units per milliliter in 25% glycerol, 50 millimolar tris HCl at a pH of 7.5, which I stored at -20 C. Dilute the enzyme with 50 millimolar ETA, pH 8, to a final concentration of about 45 units per milliliter in 300 milliliters of solution. The protocol should not be highly sensitive to this concentration, but just make sure that your cells are well digested before lysis. Vortex to resuspend the cells, then incubate at 37 C for about 1 hour until the suspension looks slightly clumpy and the optical density has noticeably decreased. Centrifuge the tubes for 2 minutes. Then pour off the supernatant. The pellets should be sticky enough to stay in the tube. Add 300 microliters of the lysis solution composed of 1% SDS and 100 millimolar tris HCl at pH 7.3. Pipette up and down until the solution is clear. If it remains cloudy, then the Lyticase digestion didn't work or was incomplete. You can increase the amount of time or use a higher concentration. Add 100 microliters of 10 molar ammonium acetate to precipitate proteins in the sample. Vortex for 20 seconds, then place on ice for 5 minutes. Centrifuge for 3 minutes. Meanwhile, add 300 microliters of isopropyl alcohol to new 1.5 milliliter tubes. A white pellet should have formed during centrifugation. Remove about 400 microliters of the supernatant, avoiding the pellet, and add to the isopropyl alcohol. Invert several times. You may be able to see threads of DNA precipitating. Then, centrifuge for 2 minutes with the hinge of the tube oriented up. You should now be able to see a pellet beneath the hinge. Pour off the supernatant. Add 300 microliters of 70% ethanol and invert several times to wash the pellet. Centrifuge again for 2 minutes with the same orientation. Pour off the ethanol, then briefly centrifuge and use a 20 or 200 microliter pipette to remove the remaining ethanol. Place the tubes on a clean surface and cover with a lab wipe. Allow the ethanol to evaporate for 15 minutes until none is visible in the tube. Add 50 microliters of rehydration solution and 1.5 microliters of 4 mg per ml RNase. You can combine these, but be careful to not contaminate surfaces with the RNase. You can also use a smaller volume of rehydration solution for more concentrated DNA. Vortex for 1 second, then incubate at 37°C for 15 minutes. Place the tubes in the fridge at 4°C overnight to rehydrate the pellet. Finally, check the concentration with a fluorometric method like qubit and quality with a spectrophotometer like a nanodrop. This DNA will be high enough quality for PCR and Illumina sequencing, but additional purification may be needed for long-read methods. Thank you for watching. Music credit to Ann Joan.